Hi everybody, welcome to Creation Myths. I'm Darwin ZDF42, I'm your host, and since this is my first official video, let me just tell you that what we're doing here is debunking science-y creationist claims. Things like today's topic, genetic entropy. I'm not going to waste time with any more introduction, we're going to get right into it. So we have to start with the question of what on earth is this thing called genetic entropy? This is an idea that was first proposed in a book called Genetic Entropy and the Mystery of the Genome by Dr. John Sanford in 2005. Sanford is a world-renowned geneticist. He's the inventor of a thing called the gene gun, which is really important for genetics technology in the 20th century. He's also a young Earth creationist, and he came up with this idea called genetic entropy. Now, the idea here is that mutations build up within a population, fitness declines, and ultimately extinction is going to be the inevitable result. Sanford wrote, and just by the way, I'm going to quote from the third edition of Genetic Entropy. So anyway, in the third edition, Sanford wrote, when selection is unable to counter the loss of information due to mutation, a situation arises called error catastrophe. If not rapidly corrected, this situation leads to the eventual death of the species, extinction. And before I go on, I want to highlight this term here, error catastrophe. This is important because what Sanford describes is basically error catastrophe, which is the accumulation of mutations over generations leading to a fitness decline within a population. Sanford was just coming up with a new word, basically, genetic entropy, for a phenomenon that had already been described mathematically. Now, the big idea with this concept, whether you call it genetic entropy or error catastrophe, is that it's really all about survival and reproduction. In effect, Sanford makes an argument by contradiction. If genetic entropy is real, evolutionary theory is fatally flawed. And the kind of carry-on to that, which I'm only going to mention again a little bit later on, just very briefly, is that if evolutionary theory is fatally flawed, then special creation is a better explanation for the existence of humanity than evolutionary processes. Now, I'm going to engage with the arguments that Dr. Sanford makes in the context of evolutionary fitness. This argument really boils down to evolutionary fitness, which correctly defined is all about reproductive success. Higher fitness means that you leave more viable offspring who themselves are capable of reproducing. One of the simplest measures of fitness is actually how many grandchildren an individual has, because that shows not only that they were able to reproduce, but that their offspring were also able to reproduce. Now, various creationists like to couch this argument in different terms. Dr. Sanford, for example, frames it in terms of information content in the genome. Now, he doesn't ever define information in a way that's quantifiable, so it doesn't really work. And I'm not going to worry about that because this argument is flawed enough without engaging with the concept of information, but that's how he frames it. Other creationists will frame it in terms of things like structural biology, like the required specificity of certain structures and the precision required in certain structures, where if they're off by just a little bit, then the function falls apart. You can actually see that form of that argument in a debate I conducted not that long ago with Sal Cordova. I'll link that debate down in the description because his argument is largely the structural biology argument. Now, none of that stuff matters because whether you're looking at information in the genome, however you define that, or precise structural features of a cell, however you want to measure those, all of that stuff is upstream of reproduction. The reason that stuff would be a problem, the way Sanford describes it, for example, is that as information declines, an organism's ability to reproduce declines. So over time, a population is unable to have enough offspring to replace itself, and it goes into a terminal decline, ultimately leading to extinction. So this is really, no matter how you define fitness, it really always comes down to reproductive success. So that's what I'm going to talk about for those reasons, in addition to the reason that that is the correct definition for fitness in evolutionary biology, and this is an argument, genetic entropy is an attempt to discredit evolutionary biology. So we're going to use the correct definition for fitness. There are a number of specific components to this argument that Sanford makes. He's very clear and he lays it out very precisely in his book. So first is that virtually all mutations are inherently harmful. And these harmful mutations must accumulate over generations. They don't slam the population all at once. They must occur over generations. And finally, this is a constant, ongoing process. In fact, it is inevitable. And I want to highlight this because Sanford picked the word entropy very purposefully. He wrote, mutational entropy appears to be so strong within large genomes that selection cannot reverse it. This makes extinction of such genomes inevitable. I have termed this fundamental problem genetic entropy, and the emphasis there is in the original. So he's on purpose drawing a parallel between the process he describes and the inevitability of the second law of thermodynamics. That is on purpose. So now let's talk about why genetic entropy is wrong. I'm going to go through the main errors as they relate to how populations actually evolve as mutations occur and compare what really happens to what Sanford claims will happen. Now, I want to be very clear about this. I'm going to describe three main problems with Sanford's idea here. Any one of them on its own is enough to invalidate the hypothesis of genetic entropy. And that's what we're doing. We're treating genetic entropy here as a legit scientific hypothesis. So let's see how it measures up. So the first problem is that fitness effects are context dependent. Sanford wrote, it can very reasonably be argued that random mutations are never good. Well, he doesn't have any data to back this up. He doesn't cite any actual data. He actually just misuses a single figure from a 1979 paper by a population geneticist named Moto Kimura. And everyone should know that name. He's one of the most important population geneticists of the 20th century. His claim to fame was on really building out this idea of neutral evolution and neutral theory very robustly. So in this 1979 paper, Kimura described a model that he used to show the importance of neutral mutations and how they can accumulate within populations. And neutral here just means any mutation, any change to the DNA sequence that doesn't have an effect on reproductive output, that doesn't affect fitness. That is the evolutionary correct definition of neutral. Now, Sanford adapts some of Kimura's figures from that paper into this figure where he shows the distribution of mutations according to Kimura. And what we see here is frequency on the y-axis and the mutation effect on fitness on the x-axis, with beneficials on the right and harmfuls on the left. And you can see this curve here. All the mutations are on the left. They're all harmful. There's literally no beneficials here. And most of the mutations are in this region where they can't be selected, which is what Kimura was trying to illustrate, that most mutations actually don't have an effect on fitness. Now, Sanford modified this. He didn't modify it, again, based on any data. He just added what he thought was appropriate. He added this teensy little curve right here for beneficial mutations. He didn't add any beneficial mutations that were selectable, that had a large enough effect for natural selection to cause them to increase in frequency within a population. But regardless, he called this the correct distribution. Now, there are some problems here. First is that Kimura explained exactly why he structured his model the way he did. And again, I want to emphasize that these were the parameters for a model, not actual data. But Sanford totally ignored that explanation. Sanford wrote, he, Kimura, obviously considered beneficial mutations so rare as to be outside of consideration. Well, that's not what Kimura wrote. In the paper itself, where Sanford got those numbers, Kimura wrote, we disregard beneficial mutants. This is an oversimplification, but a model assuming that beneficial mutations also arise at a constant rate independent of environmental changes leads to unrealistic results. In other words, in Kimura's simplified model, because he was dealing with neutral mutations, those mutations are neither selected out of the population or promoted by natural selection. So they just accumulate at an approximately constant rate. Kimura said, in my model, you can't do it any other way. And if we put beneficial mutations in there and they just constantly accumulated, well, you have runaway selection for the beneficial genotypes, and that would totally overwhelm the signal from the neutral mutants, which is what I'm trying to show. So I am purposefully going to exclude this entire class of mutations from my model. Sanford ignored that explanation. Now, it gets better or worse depending on your perspective. Again, from the same paper, Kimura wrote, this being the accumulation of mutations, this will easily be taken care of by adaptive gene substitutions that must occur from time to time. And in this context, adaptive gene substitution means beneficial mutation, mutation that improves evolutionary fitness. So what he's saying here is, yeah, you have all these mutations occurring, and some of them may be harmful, but when beneficials occur, they'll be selected for, and that solves this problem. He says directly in the paper that beneficial mutations counteract this accumulation. Well, this was pointed out to Sanford, and in 2013, he wrote, in the context of a long discussion, he said that while Kimura did not do this, include a beneficial mutation curve, I suspect he would have drawn it much as I did. Now remember that Sanford's curve includes literally zero. I shouldn't say literally zero, virtually zero, so small you can't visualize the line enough. Almost zero selectable beneficial mutations, which is exactly the opposite of what Kimura said the actual frequency would be, and Sanford has ignored that. I don't know if Sanford has not carefully read Kimura's paper or if he's deliberately representing it, but he's completely wrong on this issue, and his whole book is built on that distribution that is completely incorrect. Now, we could just stop there and call it a day, because that's it. That's the ball game as far as I'm concerned. But we can keep going. Now, the other problems in this same category are that beneficial mutations unquestionably exist, and context often matters in terms of the fitness effects of a mutation. So for example, if anyone watching this happens to be lactose intolerant, I bet you would consider the mutation called lactase persistence, which is the ability to digest lactose throughout your life, a beneficial mutation. And by the way, creationists like to claim that mutation somehow represents a loss of function or a loss of regulation or something, and it is nothing of the sort. I'm going to do a standalone video on that. Come at me. Don't even try that stuff on me. An example of where context matters for the fitness effects of mutations is the sickle cell allele in humans. If an individual has 2 copies of this allele, then they're affected by sickle cell disease, which is a very severe illness. But if they are heterozygous, which means they have 1 copy of the mutant allele and 1 copy of the typical hemoglobin allele, then they will be normal in terms of their blood cells, but they'll be somewhat resistant to malaria, which in many parts of the world is extremely beneficial. We can even look at this context dependence at the level of specific mutations and how they affect each other. This is called epistasis. And an example of this can be found in Tamiflu resistance within influenza. So in this figure, we see the fitness levels for 3 genotypes of influenza: the susceptible version, a mutant version that is resistant to influenza. It is resistant, but you suffer a fitness cost in terms of reproductive rate by having that mutation for resistance. But if a second mutation is present, that's called a compensatory mutation, it compensates for that fitness cost and actually brings you back up into the normal range. This is called epistasis, so context within the genome matters. And then finally, one more example that I really like because I don't want elephants to go extinct, that would be really sad, is the tuskless trait in elephants. Now, tuskless would be really bad if elephants were just competing with each other. But in a context where the main selective pressure, the main threat to elephants is humans hunting them for their tusks, being tuskless is a huge advantage. So we're seeing this allele for no tusks, as you can see here, spread rapidly through some populations of elephants, and it may ultimately save them from extinction. So requirement number 1, fixed negative fitness effects for mutations, that is false. I need like a stamp sound effect here, but I'm not going to do that, but that would be cool. The second problem is that harmful mutations cannot accumulate constantly. Sanford claims they do, and they must. He says, as we will see, given these numbers, there is no realistic method to halt genomic degeneration. Now, this is just simply mathematically impossible. And consider a simple case to illustrate why. If you have a genome, right? You have a genome, and you call that genome state A. That's your starting point. Now, a single mutation occurs, and now you have genome in what we can call state B. So we have a mutation from A to B. Now, once you're in state B, the mutation from A to B, and we're going to call that a harmful mutation, that mutation can no longer occur in the future because it has occurred in the past. It's already happened. But what can occur in the future? The reverse mutation from state B back to state A. And using Sanford's own logic that mutation effects have inherent fixed effects, if A to B is harmful then B back to A must necessarily be beneficial. So as mutations occur in a genome, the genome is going to approach equilibrium. And to illustrate this, we can look at this very simple figure. On the y-axis, we have mutation frequency. On the x-axis, we have number of mutations, and the red and blue lines represent the frequency of different classes of mutations. Red being deleterious. Deleterious is the technical term for harmful to fitness. It doesn't mean something got deleted, it just means harmful. And then in blue, we have beneficial plus neutral, so anything that's not going to hurt you. Now, according to Sanford, no matter how many mutations occur, the frequency of deleterious mutations stays at approximately 100%. And the beneficial and neutral mutations stay at approximately 0%. But according to math, as deleterious mutations occur, the frequency of possible future deleterious mutations must necessarily go down as each one is replaced by the reverse mutation, the back mutation, in the universe of potential future mutations. So over time, as mutations accumulate, the frequency of harmful mutations decreases, and the frequency of beneficial mutations increases until you reach a dynamic equilibrium where both classes of mutations are happening at approximately the same rate. Now, I want to be very clear here. This is not an evolution problem. This is not a genetics problem. This is a math problem with Sanford's argument, and there's no way around this. If you follow Sanford's logic of fixed fitness effects and every mutation being deleterious at the start of wherever you choose to start your analysis, this equilibrium must ultimately be the result. So requirement number 2, harmful mutations accumulate constantly. That is also false. Requirement number 3 is that this mutation accumulation must occur over generations. Sanford actually puts a number to this. He doesn't say the word linear accumulation, but he describes an approximately linear accumulation of mutations to the tune of at least 3 additional deleterious mutations per person per generation. And he's also very clear that selection is unable to remove mutations from the population. This necessarily means there is variation within populations, because mutations are random and not everyone is going to get the same random mutations every generation. This means that within a population, some individuals are going to be less bad than others. So we can follow Sanford's logic. From a starting point, mutations occur and they accumulate within a population. Individuals get different mixes of mutations over time, over generations. So within a population, some individuals necessarily are going to have mutations that are less bad than what other individuals have. And once you have that variation in a population, something is inevitable. And that something is mutation selection balance. In this figure, and we're going to illustrate what Sanford claims happens and then compare that to what actually happens in populations. In these 2 graphs here that we're going to draw, mutations are on the y-axis. So you go from 0 mutations to a lot of mutations. And across the x-axis is generations. Now, what Sanford claims happens is that within a population, mutations accumulate over time, right? Whatever your starting point is, everybody gets mutations until you hit a threshold. The dotted line represents the kind of viability threshold, where once you cross that threshold, you have too many mutations, now you're no longer viable, and your rate of reproduction is going to drop off. Eventually, it's going to hit 0, and eventually everyone's just going to be so low fitness they're going to die without reproducing. Okay, so once you hit that threshold, according to Sanford, because everyone's getting mutations and you can't select them out, they just keep accumulating, even when there's a fitness cost associated with those mutations. That's according to Sanford. But what actually happens is that as mutations occur in a population, what we might call tolerable mutations are going to accumulate. Mutations that don't immediately kill an individual or prevent that individual from reproducing accumulate within the population. That increases the variation. That's why I've drawn this as a wedge instead of a line to signify that variation in mutation load among individuals in that population. Now, you don't just magically stop when you hit the threshold level of mutations. Mutations are going to keep accumulating, and some individuals are going to cross that threshold. They're going to get too many mutations, or they're going to get specific mutations that are what we might call intolerable, mutations that prevent them from reproducing, or in the most severe cases, just kill them outright. Now, in those cases, those mutations are going to be selected out of the population by definition because the individuals with them are not reproducing. So that kind of shifts the population back down below that threshold, and that's what's going to happen indefinitely once you hit that point. You kind of hang out at a dynamic equilibrium right at that threshold of viability. This is called mutation selection balance. It's when new mutations are entering the gene pool of a population about at the same rate that existing mutations or those new mutations are being selected out. So on net, you don't have an accumulation of mutations over generations. Now that's all kind of theoretical, but we don't have to stop there because there's direct experimental observation of this exact phenomenon. And my favorite example of it is a paper by Springman et al. From 2010 called Evolution at a High Imposed Mutation Rate, Adaptation Obscures the Load. In this experiment, the researchers treated a bacteriophage, a virus called T7, with a mutagen and caused its genome to become saturated with mutations. This just means that within the T7 population, all mutations were present. Every possible single nucleotide mutation was present in that genome. And in that paper, they actually document the mutation load per virus, and it's anywhere from a dozen or more up to over 100 mutations per viral genome. So every individual in this population is getting slammed with tons of mutations. They then directly measured fitness in terms of growth rate. And this is a great test of Sanford's hypothesis, because according to Sanford, remember, all mutations are inherently harmful, and they accumulate constantly. You can't get rid of them because they're mostly harmful, almost all of them. So if a population experiences every mutation many times over, according to Sanford, that population must inevitably go extinct. But that's not what happened here. In this experiment, the average fitness declined, but the maximum fitness they observed actually increased. And this is explained by the occurrence of beneficial mutations. And in that paper, they actually document specific mutations that were selected for, and the occurrence of that mutation selection balancing point that I just described a few minutes ago. So what happened here is within this viral population, everyone's getting slammed by mutations. Most of them are not fatal, but they're not good either. But sometimes you find a really good one, and that helps you. Because you're just generating tons of mutants all the time, even those really high fitness genotypes are constantly being bombarded with more mutagen, and they're mutating away from that fitness peak. But you're also constantly generating those high fitness genotypes, which are being selected for and therefore maintained within that population. That is impossible according to Sanford. So this experiment is a direct refutation of the genetic entropy hypothesis. So requirement number 3, no selection over generations. That is also false. The last objection I have, and I'm not going to spend too much time on this, but it does matter, is that genetic entropy is an example of the begging the question fallacy. And this means that it sneaks the conclusion into the premise. And Dr. Sanford does this by assuming optimal genomes at the start, which subsequently accumulate mutations and degenerate. Evolution doesn't work like that. In evolutionary theory, there never was a perfect mutation-free genome. But you know what does work like that? Special creation involves a perfect starting state followed by degeneration from that ideal. By framing his argument in terms of this optimal starting genome, Sanford assumes his desired conclusion, special creation, from the start. That alone is enough to invalidate his entire argument. I want to focus on the biology, and that's what we're going to keep talking about, but in terms of this litany of problems with his idea, this has to be mentioned because it alone invalidates the idea. So now let's talk about the observed evidence. I'll start by saying very clearly that genetic entropy has never been observed. A 2012 paper was published by Dr. Sanford and Dr. Rob Carter on H1N1, and that paper is very frequently invoked as an example of genetic entropy. If you ask a reasonably well-informed creationist, reasonably well-informed in this debate, for an example of genetic entropy, 9 times out of 10, you're going to get this paper. This study was awful, frankly. It had tons of flaws. They did not actually document genetic entropy, and they claimed an extinction event and then subsequently refuted themselves. Now, I'm not going to go into the details here because down the road, I'm going to have a standalone video on this paper. That's how bad it is. So keep an eye out for that down the road. But for now, suffice it to say that this 2012 paper that is often used as an example of genetic entropy did nothing of the sort. There was also a 2006 review called Examining the Theory of Error Catastrophe. And remember from before, Sanford basically stated outright that genetic entropy is error catastrophe. And in that, again, this 2006 review, the authors wrote, "We find that in general, experimental support for error catastrophe is marred by the failure to propose or test alternative explanations for the results and by inadequate precision in the data." And I can say from my own experience that the literature since then has not been any kinder to the idea of error catastrophe. There are no good examples where we can confidently say this is error catastrophe observed in an experimental population. There are also direct refutations of Dr. Sanford's hypothesis. Now, we've already talked about an experimental example, Springman et al. 2010, but we can also look to natural populations, because if Sanford is right, some species should 100% be extinct, but aren't. My two favorite examples of this are HIV and mice, because let's talk about mammals too, in case you don't want to talk about viruses, you being a creationist in this example. So starting with HIV, early in the HIV pandemic, you have the exact situation that Sanford describes in his book. He says, inbreeding and genetic drift then take over entirely, rapidly finishing off the genome. The process becomes an irreversible downward spiral. And what he's describing there is a thing called an extinction vortex. And it's where you have a small population that suffers from inbreeding and genetic drift. Genetic drift is random changes in the gene pool of a population, and genetic drift is dangerous because since it's random, harmful mutations can accumulate within that population. And inbreeding is dangerous because you can end up with lots of recessive disorders that are actually expressed because you end up with two copies in an individual more frequently than you would otherwise expect. This situation describes HIV early in its pandemic. HIV, human immunodeficiency virus, evolved from a virus called SIV, simian immunodeficiency virus, specifically the chimpanzee variant. I'm talking about HIV-1 group M, which is the pandemic strain, if you want to get into the specifics there. So what happened here is in the early 20th century, 1920s, 1930s, a very small little bit of that SIV population jumped into humans and became HIV. Now that was a very small and very not genetically diverse population. In other words, it's exactly the situation that Sanford describes here. It's susceptible to an extinction vortex. In fact, HIV is uniquely susceptible because it has an extremely high observed mutation rate because it's a retrovirus. It would have had very low genetic diversity, high drift because it would have been a very small population initially. So you put all those things together, and HIV should have gone extinct at some point after that crossover, but before it exploded into a pandemic. Now, the crossover event happened in the 1920s or 1930s. The pandemic spread didn't start probably until during the 1960s into the 1970s, and we didn't notice it until 1981, and that's when, kind of officially we consider the HIV pandemic having started. So in those intervening decades between like 1930 and 1960, if Sanford is right, HIV should have gone extinct. Mutation should have just built up, and then it should have just fallen off a cliff. But that obviously didn't happen, and that is a direct refutation of genetic entropy. Mice are kind of in a similar situation. Mice have the same mutation rate as humans, and mutation rate is described as mutations per site per replication. That's the technical way to measure mutation rate. That's from a 2017 paper. We have the same mutation rate as us. And they should have a higher substitution rate. That's substitutions per site per year, so changes to locations in your genome per year. And this is the case because mice have a generation time of about 10 weeks, whereas humans have generations of 20 years or more. So over many years, mice should be accumulating more mutations in their genome than humans. And Sanford is very clear that humans are experiencing genetic entropy. So if humans are doing it, then certainly we should be able to see evidence of it in mice. Well, the weird thing is that their substitution rates are approximately equal. And in fact, mice are a little bit lower than humans. And this is according to a 2019 paper. They're about the same order of magnitude, but mice are actually a little bit lower here. That should not be the case based on what we know about mice mutation rates and reproductive rates. So why in the world do they have a lower substitution rate, which is to say slower mutation accumulation? Well, the answer is obviously selection. They have an enormous population and fast generation times, which facilitates much more rapid, or I should say much more efficient selection compared to humans. So that explains what's going on there. And that's a direct refutation of the genetic entropy hypothesis, because according to Sanford, selection cannot act on these mutations as they accumulate. And if that was the case, then mice should show more mutation load than humans, but they don't. And that's a direct refutation. So to summarize all of this, genetic entropy is population extinction due to mutation accumulation and loss of fitness over generations. It is flawed because, first and foremost, it begs the question. It sneaks the conclusion into the foundation of the argument by assuming an optimal starting genome. Evolution doesn't work that way, but creation does, and that alone invalidates the argument. But, sticking with the biology, fitness effects are context dependent, and beneficial mutations exist. Harmful mutations cannot accumulate indefinitely in populations, and again, that's just basic math. And populations with variation achieve mutation selection balance. Selection occurs on that population, so mutations don't accumulate infinitely. You end up being selected out if you cross that threshold I represented earlier with the simple graph. And then finally, we just have direct experimental and natural refutations. There are experiments where those populations should have gone extinct if Sanford is right, and there are natural populations that should be extinct if Sanford is right. The fact that we didn't observe those things is a direct refutation that demonstrates that the genetic entropy hypothesis is without merit. So, genetic entropy, our first creation myth that we discuss in this channel, that is definitely a myth. And that is where we are going to leave it for today. Thank you for watching. I hope you enjoyed. If you enjoyed this video, please give me a subscribe, comment, give me a thumbs up, whatever people do to get these videos a little more visibility. Obviously, I'm just starting out, so I would appreciate whatever you want to do in that regard. And if you watched this video and hated this video, you know what? Give me a hate comment, give me a hate share, whatever you want to do. Just spread it far and wide. Do me a favor. Thank you. So I hope you all enjoyed this. And what I'm planning to do is do things like this regularly-ish. We'll see what the schedule ends up being. But until next time, thank you for watching. Remember, always check creationist sources and don't get fooled. See you next time.